From Microbe TV, this is Immune, episode number 88. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, everybody. Welcome back. How are you doing, Vincent?
Okay. Is it snowy up there yet?
Well, we did have snow, and then it rained, and all the snow went away, and now it's really warm today, and I made a bad choice because I wore a sweater, and I'm sweating.
Yeah, it's been like minus 5°C here for a couple of weeks, and now today it's 5, and it's raining. But Friday, it's going to go back to the sub-zeros.
Yeah.
Like -9 and -7. It's ridiculous. Also joining us.
It is winter, so, you know.
What? It's winter?
Actually, technically not for a couple more days, right? Isn't it December 21st is the actual official date? Yeah.
Also joining us from Cleveland, Ohio, Steph Langle.
Hi there. Hi, everyone. It's cold here. Well, no, I'm sorry. It is going to get cold here. It's warm right now. All that snow has melted. We live east of Cleveland, which is where the lake effect snow really drops. We had 17 inches a couple weeks ago, but it's gone.
Did you see the dump that they got in Buffalo, or it wasn't Buffalo?
We didn't have it as bad as
all that lake effect snow. They got like 2 feet per hour or something.
Buffalo is problematic. Don't go to Buffalo.
Poor Buffalo people.
Also joining us from Madison, New Jersey, Brienne Barker.
Hi. I happily do not have any of that snow. It's actually 14 Celsius here, but that is the sort of northern tier, Tug Hill, near where my parents live. So yes, I know exactly which snow you're talking about, Cindy.
Yeah, it was a lot, right? Yeah. It was almost a record per hour drop. I can't remember how much it was.
So Steph, you were in Uganda recently?
Yes, I was for a week. It was my first time to the continent of Africa. And Case Western has a relationship with Makerere University, which is in Kampala. And it was an incredible experience. I saw many things. And listen, if you want to study infectious diseases in maternal and neonatal populations, that would be a place to do it. So it was, quite life-changing. I think we're going to be setting up a cohort there. So it was great.
Cool.
Thanks for asking.
Well, if you like these programs where we talk about real science, not fake science, not lies. Did you see that RFK wants to get rid of the polio vaccine?
It's insane. It's terrifying.
This is just beyond normalcy. This is just stupidity. I don't get it.
I don't get it either.
Anyway, if you like what we do, you should help us, support us, because we depend on your contributions. Microbe.tv/contribute. Yeah, it's just crazy. It gets crazier and crazier every day.
Yeah, I think the outcome of successful vaccines are a lack of the disease that the vaccine causes. It's interesting when you look at, various message boards, depending what platform you're looking at, there are people in, 70s who remember a sibling got polio. They remember their polio vaccine experience. And so, the goal is so that we don't have to repeat that. But unfortunately.
No, it's funny. Mitch McConnell, who had polio, said, you're not getting rid of polio vaccine. It's a good one. So did Trump. He said, no, I think polio vaccine is a good one. In fact, they're all good.
They're all good. And Steph is exactly right about sort of people forgetting the importance of vaccines. And I have been struck by how quickly that seemed to happen, because these stories of people who had polio are of people who are still alive, or like people we know, or, grandparents or something. It's not as if this has happened over so many generations that we've had time to forget. We forgot in like a generation.
I just don't think we need to get kids getting sick and dying to make people realize that they need to get vaccinated. No. This is crazy.
Yeah. I wrote about this. So I wrote an opinion article back in 2015. It's almost 10 years.
Oh, wow.
We're still talking about this. Yeah, it got published on CNN. It's called What We've Forgotten About the Danger of Childhood Vaccines. But it was exactly that idea. That's a societal amnesia. Exactly what you said, Steph, is that, we don't see those diseases anymore. So people are making their own risk analysis and saying that the vaccine is more dangerous than the disease because they've never seen the disease and they have no concept of how dangerous getting the disease actually is.
Yeah, it just means we get our jobs as science communicators.
Yes,
are not over.
Correct.
For sure.
Right.
That's why you need to support us. Microbe.tv/contribute.
Yes.
I saw recently someone saying, "Oh, I'm not against vaccines. I just think we give kids too many at once. It's too much." And I'm like, in a single infection, you get way more antigen than in your
a lot more proteins,
hexavalent vaccines. Come on, don't be silly.
Sitting under an air vent or being on the subway or living your daily life, you also probably see more microbes.
It's just a
But maybe this is an opportunity to think about next generation pediatric vaccines. Can we combine antigens so that parents aren't feeling that it's so many doses?
We do have these hexavalent vaccines, right?
Yeah.
Which include polio, and the world should use those because they're all injected at once, and that's when people say it's too many antigens. So they've combined them so people don't have to go to the pediatrician too many times, and now they say it's too many antigens.
Right,
it's just you cannot win. I just don't get it. There are many countries, including Uganda, who would love to have all the vaccines we have.
Listen,
right?
the amount of infectious disease that I saw, and they don't have, they can't afford a pneumococcal vaccine or, influenza vaccines. I believe rotavirus, because it's cheap, is accessible, but not in the really low poverty areas. And there's a malaria vaccine they're trying to roll out. I mean, so many kids die of malaria. So absolutely.
Yeah, maybe we need to have a program in the U.S. where if you don't want to immunize your kids, you donate that vaccine to a country that needs it.
That's a great idea.
And then you see what happens.
Well, what's going to happen is the rest of the world is going to get healthier and our children are going to get more sick.
Yes, indeed.
Unfortunately.
All right. Anyway, go to microbe.tv/contribute and give us what you can so that we can keep talking about science and debunking things and so forth. Today we have a snippet and a paper for you, and I will tell you about an interesting study in bats. This is a paper in Cell Reports, application of a bespoke. I have to tell you, bespoke got me,
I did. I had to look it up.
Don't put SAT words in the title of a paper.
Bespoke. Well, you go to Hong Kong to get a bespoke suit,
Yeah.
Tailored to you.
Tailored to you.
Well, this is part of the problem of science, right? You communicate, you put that kind of thing in a title, and they're like, that is the intellectual elite that doesn't want us to understand what they're talking about.
I will say that every time I think I've heard the word bespoke before this paper had to do with clothes.
with clothes.
Yeah, this is the first time I've seen it on a science paper.
It's clever. I mean, I'm not saying that it's not a word that can't be used, but if we're really trying to communicate to a broader audience.
I don't know if you even need bespoke. I mean, you're making monoclonals against bat protein. Anyway, the title is Application of a Bespoke Monoclonal Antibody Panel to Characterize Immune Cell Populations in Cave Nectar Bats. So this comes from the Duke National University of Singapore Medical School and the National University of Singapore, both in Singapore. And many authors here. The first is Shiwei Chen, and the last is Linfa Wang. I've been to Singapore. I've been to this place, Batman, and did multiple podcasts with Linfa. So, everyone should know that bats are unique in many ways. They're flying mammals. They constitute 20% of all mammal species.
That's a huge amount. I hear that stat, and I think.
But rodents are 40%.
Right, and not to be beat out.
Wow.
But they don't fly.
They don't.
Right, fair. Flying rodents.
Thank goodness.
But humans do fly, and we make a mess of things, right?
Slightly different metabolic costs.
As we've learned in the last 20 years, bats harbor many viruses, many which are pathogenic for humans. Many spill over into people like Nipah virus and Hendra and others, coronaviruses. And they seem to be okay with many infections. So people are interested in why this is the case. Why can they handle the infection? And we have some information from a variety of studies. For example, they seem to be good at dampening their interferon responses, so we don't get over inflammation. And also, they fly, and that generates a lot of oxygen radicals. So they do have interesting adaptations to reduce inflammation caused by oxidation. So maybe that spills over from flying into infectious diseases.
Yeah, basically
as well.
They seem to react to certain nucleic acids less, and that might have evolved to not respond to nucleic acids that are being made during all of these DNA damage processes coming from metabolism, but has sort of the side benefit for them of letting them not have pathology with these viral infections.
We talked about this on a prior immune, right? I looked it up. Immune 46, bats don't sting like a bee, which is interesting because we're going to talk about sting later. Yeah. But yeah, they have dampened sting responses. So their inflammation to the virus is lower.
I love this stuff.
Bats are non-model organisms, right? So what does that mean that you're a non-model? So there are not a lot of reagents.
I was going to say they don't have the reagents.
But do they have to be inbred to be a model organism? I was thinking of it this morning.
So. I don't think they have to be inbred to be a model organism.
Yeah, I didn't think so.
Because non-human primates, they capture them, right?
Yeah, ferrets are not inbred, right?
Right.
So just that you have a lot of reagents. And mice are basically one species, right? Musculus. But there are lots of different bats. And so which one you're going to pick if you want to make one a model organism. So that's what this paper addresses. They have a breeding colony of the cave nectar bat, Eonycturus speciale, and we'll call that ES from now on. They're nectarivorous bats, so they don't eat insects, they eat nectar. And they are classified as a least concerned species under the International Union for Conservation of Nectar. The genome has been sequenced, and there's been single-cell transcriptomics done, and so forth.
That's already so much more than some of the other bat species.
Yeah.
Yep.
Yeah. Anyway, so in this paper, they create a panel of monoclonal antibodies to immune cell markers. When we talk here on Immune about papers, they're always using antibodies against markers to characterize specific cell types, like CD4 and CD8 and many, many others. So they make a panel of monoclonals here, and they do some characterization of those, which bring up some interesting results. So what they do is they pick 7 immune receptors, and they make soluble forms of the protein. They immunize mice with them, and then they take the B cells, they sort out the B cells with recombinant protein. They do single-cell sequencing, and they find the sequence of the B cell receptors, and they clone them and make antibodies.
Yeah, I thought that was a cool way of doing it.
All right, and so they say that so the CD45 is a marker that marks pan-hematopoietic cells, and so they have an antibody that they made that they think is similar, SX52, and they do a number of studies to show that in fact SX52 is a B cell marker that's similar to CD45, which we've also called B220 many times, right? So that's, they're going to call it CD45RA. And then they have some other monoclonals against other proteins as well. So what they do now is they take some major organs from the bats. They take spleen, blood, bone marrow, lung, and intestinal tract, and they do some flow cytometry to characterize the presence of cells that react with their antibodies. And by the way, just this, I found this very interesting. The bats are heavier than mice. So they do a lot of comparisons with mice in this paper. So they're
a lot bigger.
They're 2.7-fold heavier. So they're 53.2 grams on average compared to 19.6 for mice, musculus, right? All right, so in the spleen, B and T cells are most of the immune cells, but they found that the B cells are much less in the bat compared to the mice. And then the rest of the cells are innate immune cells, NK-like cells, granulocytes, and monocytes. And they find that monocytes between the bat and the mouse exhibit the biggest difference in numbers and phenotypes. The bats had more monocytes, and these were equally divided between classical and non-classical monocytes. And in contrast, the mouse has primarily classical inflammatory monocytes with fewer non-classical patrolling monocytes. In the blood, there were fewer lymphocytes in the bat compared to the mouse. So, for example, B cells and T cells, both CD4 and CD8 cells, 22 to 32% of the lymphocytes in the blood, compared with 48 to 63%. And the biggest difference is in the CD4 positive T cells.
So I wondered a little bit about some of these numbers and comparisons to mice. And I sort of can't tell from the methods exactly some of these details. But I know that there's a paper I always talk about with my immunology class that talks about how mice are kept in specific pathogen-free housing. And the fact that that might change their exposure to pathogens and their sort of baseline immune parameters. And so I wondered, because there are so few bat colonies, are the bats kept in the same kind of specific pathogen-free environment? Are the bats sort of in association with more microbes than the mice? And how does that comparison made? And I don't know enough about these bat enclosures to really be able to sort of tell that difference.
But it's a good point. We talk in a lot of effects of the microbiome in mouse studies and having to have caged littermate controls. And so for sure it matters, and it would be interesting. Can you make a germ-free bat?
Well, even an SPF bat.
An SPF, sure.
Because if you think about some of the other non-model organisms, in some cases they are kept in much different types of facilities. Here they mention, that they have wire mesh to prevent the entry of outside animals. But that's very different than if you think about how we go into a mouse facility with a gown and masks and hairnets and all of that. What kind of microbes can be brought in? So I thought that was a really interesting thing. I don't know how you entirely address it.
Be interesting when you think about the bat colonies, quote unquote, at zoos. I'm just thinking of the zoo that I'm familiar with. It's not open to the outside like this. So I wonder if they could compare.
I just found a paper that was published in 2022. It says establishment of a captive cave nectar bat breeding colony in Singapore. It's Lim Fa Wang. And it says that they trap bats were removed from this nets, placed into custom-made, breathable, opaque cotton mesh holding bags, and then weighed. And bat mites were removed using blunt end forceps. Any physical wounds were treated with antiseptic cream. They were quarantined for a month before being introduced into a colony. All personnel wore Tyvek suits, safety goggles, N95, headlamp, and bite-resistant leather gloves over nitrile gloves when trapping bats. And then it does say bat facility included air-conditioned anteroom connected to a BSL-1 lab that is connected to the bat holding area. It's sheltered, naturally ventilated, exposed to natural photoperiod, and enclosed by wire mesh to prevent entry of rodents, birds, and wild animals. But I'm looking at it as like a big box. So I think probably that, other bats and animals could like land on top and probably share fluids.
Feces.
Sure.
Urine, right.
Yeah.
Yeah, so that could account for a lot of these differences. Yeah.
Yeah.
Which is something they could deal with in some future study. Bone marrow Most of the cells are granulocytes, similar to mice and bats. Again, for the bats, lower numbers of B cells. Lungs. So, big differences in the proportions of cell types, immune cell types between lung and the blood. The bat lungs had equal numbers of B cells, CD4 and CD8 T cells, and granulocytes, whereas in mice, B cells are clearly the dominant cell type, 1/3 of the immune cells. And the mice also had higher numbers of NK cells, but that was compared to the bats. Okay, intestinal tract. So they have short intestinal tracts. I didn't know this.
I didn't know this either.
So the intestinal tract of the bat is only 12% longer compared to the mouse, despite the bat weighing 170% more. Interesting. The intestinal tract has a smooth outer appearance with no morphological differences between the large and small intestines, and no Peyer's patches.
I was surprised by that. That's very interesting.
It's quite interesting. So that's not something that is caused by the environment, I think, right?
Well, mice develop them after birth when they're exposed to microbes.
Yeah.
So if anything, you would have expected the bats to have potentially more Peyer's patches.
We just said they are exposed. Yeah.
But you're saying in the bats, exposure isn't.
Yeah.
It's not. That's not.
Well, they have no Peyer's patches at all,
right.
which is interesting.
Right.
So they think there are 3 segments in the bat intestinal tract: the duodenum, jejunum, and then 2 segments of ileum and the colon rectum. They what they did is to digest cells from different parts of the tract and stain them with their antibodies. They couldn't do this with mice, so they couldn't compare them. So they just had to report what they found in the bat. So the B cells are mainly found in the ileum. And the start of the ileum is almost completely devoid of B cells. T cells, however, are found in all the sections that they looked at. So they say, okay, why are there no B cells in parts of the gut? Maybe our antibodies are not working for those particular markers. So they did some Swiss roll histological sectioning, which roll up the intestine, and then they make sections.
Annoying to do.
We talked about that on Twitter a long time ago with norovirus and my Stephanie Karst. And then you can look at instead of having to take sections of a very long intestinal tract, you got it all in one roll, right? And you can just see it. So then you can stain it. You can also look at morphology. And so despite lack of staining, they could see plasma cells in these intestines. So throughout the intestine, there are plasma cells, but they didn't stain with their markers. So that's an interesting point in that, their antibodies aren't perfect.
Right.
Yeah. So then the last bit is, can we use these for other bats? Is there any cross-reactivity? So they have splenocytes from the black flying fox and the lesser short-nosed fruit bat, and they check them for reactivity. And only 2 of their 5 monoclonals cross-react. So it's too bad, right? Because you'd like to be able to do this in multiple bat species, but they just don't cross-react. They're different enough.
And that's really interesting because these 2 species they used are sort of closely related. In the full diversity of bats, there are bats that are much more distantly related. And so if it's a small fraction of the antibodies are working, even in these closely related bats, that is really sort of unfortunate that it's going to make this harder. But also really fascinating, perhaps, that bat immune systems are evolving that quickly.
Yep.
Do you guys know how cross-reactive the antibodies we normally have for mouse are with the other mouse species, like Spreadus and
Peromyscus?
I imagine they are. Just considering we use mouse antibodies, they sometimes cross-react with human, or we're using them to see if they cross-react with ferrets, which are really closer to dogs. Actually, the dog antibodies work the best. So probably there's conserved, but that would be interesting if it wasn't.
Okay, so that's it basically. I want to point out two things from the discussion. First, they say, this finding that there are fewer B cells in the bats compared to the mouse in all the compartments they looked at. It's several people have suggested that humoral immunity is not a big deal in bats because there's not a lot of seroconversion and non-neutralizing, and a lot of the antibodies are non-neutralizing. So maybe he's thinking, well, maybe there's not a lot of B cells either. Maybe that could account for that. And they also say that this finding that non-classical monocytes are really more common in the bat than the mouse. Maybe they are playing a role in limiting pathological damage during virus infection. So obviously you could do some experiments to address that. So there you go.
I really like coming back just to the very beginning, how they made these antibodies. I thought that was very clever. We're reading it all the time now in papers where people we did one, I think last month, where they were looking at B cell responses and they just clone all the BCRs and then they make them and then they test them. So it's becoming kind of commonplace, and it is cutting down the amount of time that it takes to make monoclonal antibodies from what we had to do before, which was immunize a mouse and then take the spleen and then crush it up and fuse it, make hybridoma, and then collect the screen. So it's really interesting to be able to do the single cell paired heavy and light chain sequencing to find out the active antibody. I
remember the old days when we used to make hybridomas against viruses. You would immunize a mouse and you'd wait and you'd take out the spleen and you'd fuse it with another cell line, right? And then you'd put them in 96 well plates.
Yep. And grow
hundreds
them all
and hundreds. And then screen them,
yep.
Like with a neutralization assay. But here you use the recombinant protein to pull out the right B cells. And there you go. It's great.
I no longer have to make my students learn the way I did the components of hat media.
It's the way it should be in science, that things change, right?
Yeah.
Although I love talking about old techniques. They're great.
Yep.
We did a paper on recently where they were doing thin layer chromatography.
Oh, wow.
Guys remember that? No, probably not. I don't know.
Yeah. I mean, not that I did it, but
when I was a student, man, there was a lab. It was scary because some of them were driven by incredibly high voltages. Oh my, it's scary. You could kill yourself.
With a lot of radioactivity too.
Safer now.
Well, yeah, a lot of radioactivity. That's right. In this paper, they used radioactivity to see the products. Anyway, now on to Steph.
Great. Well, thanks for that snippet. It was really enjoyable. The next paper we're going to talk about is a paper entitled Retrotransposons are co-opted to activate hematopoietic stem cells and erythropoiesis. And the first author, I don't believe there's shared authorship. I'm just seeing Julia Fan. And there's co-senior authors, Al Paslin, Tez Doggan, and Sean Morrison. Sean Morrison is at the Children's Research Institute at University of Texas Southwestern Medical Center, and the senior author is from the University of Duisburg. Not sure if I pronounced that. It's in Germany, Essen, Germany. And this paper, I thought it hit upon many interests amongst the group. So I think it'll be fun to talk about. There's questions I have. I'm excited for your feedback. Before we start, I want to give a little vignettes on the different components of the title, which will really help us frame the paper. So firstly, hematopoietic stem cells, or HSCs. What are they? They are multipotent stem cells. They're located mostly in the bone marrow, but you can find them in other places. Think the umbilical cord, fetal liver, fetal yolk sac. You can find some in the spleen, which will be relevant for this paper, and various other organs, particularly under stress conditions. Why they're special is that they give rise to all blood and immune cells through hematopoiesis. And they have this unique ability to self-renew. They maintain themselves in this stem cell pool, but they can, as I mentioned, differentiate into myeloid lineage cells, which are red blood cells, platelets, neutrophils, or lymphoid lineage cells, which are T cells, B cells, and K cells. And they're really relevant for regenerative medicine because of this ability to produce identical daughter cells that retain stem-like properties, and they have specialized niches depending on where they're located. So you can imagine they would be important for stem cell transplants to treat a variety of diseases: leukemia, lymphoma, anemia, sickle cell anemia. But you can take them out of the person and modify them ex vivo to correct for inherited defects or enhance the function and then reintroduce them into the patient. So there is even long-term benefits to those type of therapies. You can induce hematopoiesis under certain stressors: infection, blood loss, anemia, radiation, chemotherapy, transplantation. And what we're going to talk about in this paper, pregnancy, may also be one of these stressors. And so the group frames the paper around a previous paper they had published in 2014, where they demonstrated during pregnancy that estrogen or estrogen receptor signaling activated these HSCs, hematopoietic stem cells, increased their cell division, and something called extramedullary erythropoiesis, or in simpler terms, making more blood cells in the spleen, which is an organ that doesn't bear the brunt of most red blood cell production, although it does filter them. And so they were seemingly following up on this to really understand the mechanism of what was going on in that previous work. So the next thing we need to talk about in the title are... go ahead.
Well,
Yes.
I was just going to say, even from the beginning, when I was reading the information about pregnancy influencing hematopoiesis and particularly erythropoiesis and sort of the importance of that, I was like, I had never thought about that. And of course that's important. Like, of course that would have to be a thing. And yet it didn't occur to me until reading the intro of this paper.
Yeah. Yes. And it would be detrimental to the species if you became anemic and did not increase your blood flow, considering the placenta is highly vascularized, the baby needs its own blood. So there has to be a way for the mother. I mean, and typically, during pregnancy visits, they will track you to make sure you don't become anemic. It's not impossible that you could become anemic, and iron supplementation is common in prenatal care, but that you wouldn't become so anemic that the pregnancy wouldn't survive. So yeah, it's a great point, Brianne. It's kind of like you think about these things that are just known knowns, and you're like, oh my gosh, this is actually quite stunning. So the other concept is these things called retrotransposons. And so what are retrotransposons? It's germline DNA elements that are capable of moving from one location to another. So germline refers to the genetic material that's involved in reproduction. This is sperm and egg cells, as well as whatever cells the precursor cells were that became them. So they are involved in reproduction. They're passing to the next generation. And these retrotransposons, they do this copy and paste mechanism by first being transcribed into RNA, then reverse transcribed into DNA, and then inserted into a new location in the genome. And they're doing this using the enzyme reverse transcriptase that they themselves encode, which is pretty incredible. So the activation of retrotransposons would then lead to this type of activity. And if you've heard of things like endogenous retroviruses or ERVs, they can do this. Lines, which are long interspersed nuclear elements, they can produce reverse transcriptase. In the paper, they include signs within the group of elements that can produce reverse transcriptase. But if I'm correct, that actually isn't true. The signs don't make their own. They co-opt reverse transcriptase from lines and ERVs. So that was just a comment.
But either way, you could have nucleic acid made.
Right. And so it would make sense that this activity is repressed because this can cause genome instability. You can cause transposon-mediated mutagenesis. These are things you don't randomly want your germline changing genes and jumping from one place to another. And so you can imagine there are things in the cell that can detect when you have production of DNA, RNA, or reverse transcribed DNA that are produced as a result of transposable element transcription. And these are called innate immune sensors, which 50% of the group have labs based on these molecules. And these are things, you've probably heard them on the show before, toll-like receptors. But there's a couple that we're going to talk about in this paper. There's many, and this is a whole exciting, burgeoning field of detectors in the cell. But cyclic guanosine 3,5 monophosphate, adenosine 5 monophosphate synthase, and I had to write that out for myself because I did not memorize that for people listening. That was read. C-gas is what we're going to call it. And then what C-gas can stimulate is something called STING. And STING, we've talked about a lot on the show, stimulator of interferon genes. Interferon genes are pro-inflammatory molecules that help cells communicate. So if you are a hematopoietic stem cell, there are times when if you encode these ERVs lines, it would be beneficial for you to upregulate them. That would induce activation of yourself as a cell. This could be aging, radiation, chemotherapy, DNA damage, and as we discussed, pregnancy if you have to make a lot more red blood cells.
And so that might be a reason why we keep the retrotransposons, even though they have some potentially negative consequences. There's some downsides to them. And so it's a little weird. Why have we kept all of them around in some cases? And perhaps it's because we're going to use them in this kind of way. Right.
It would make sense that we would keep them around. You're right. If they are able to do key functions, while under most conditions, we would want them repressed so that we don't. Yeah.
And I wanted to add to that because there was a cool paper, and I looked it up while you were talking, that came out in 2012 from Stefan Bauer's lab. And the paper basically said that if you delete the nucleic acid sensing TLRs, so like all of them, the endogenous retroviruses start hopping out of the genome and being activated. So their argument is that the nucleic acid sensing TLRs, one of their key roles is to keep those endogenous retroviruses in check. So that kind of like got my head spinning, because here is a situation where you actually want to activate them to be using them for something. So I've always wondered, what is there another role for the nucleic acid sensing TLRs other than detecting virus nucleic acids? Because it's not, you delete them and you can show unique susceptibilities to certain viruses under certain conditions, but it's not like you have a global susceptibility to viruses when you delete a lot of the TLRs. And so I've always wondered, is there something else going on?
Yeah, I saw a talk and have since read a paper by from the Belcade lab that was talking about similar things, that some of these retrotransposons may help us have sort of a basal level of certain cytokines to help protect us early from viral infection. It was a fascinating paper, and also an incredibly detailed and deep, dense paper, because I've thought about doing it in immune or in classes, and it feels like it has about 5 papers worth of stuff in it.
That's going to be a 3-hour-long episode.
Yeah. So, but that was one thing I know that's really cool about that, is the idea that that we may have taken advantage of this also for sort of tonic background levels of cytokine.
So cool. I think it's really an exciting area of study for many reasons, and it really framed the start of this paper defining the mechanisms of retrotransposon-induced hematopoietic stem cell activation, particularly during pregnancy, and they'll talk a little bit as it relates to estrogen signaling. So the paper starts off by looking at transcriptional changes, single cell RNA sequencing, in hematopoietic stem cells. They also look at hematopoietic stem cells within the bone marrow and spleens of either pregnant or non-pregnant mice. Now, what they did that's different than what most people probably do with RNA sequencing, for sure, what I've done in the past, is they included the coding sequences of DNA that can contain the instructions to make protein. That's messenger RNA. But they also included what typically gets thrown out is the transposable elements that are not usually in the exons. And so including both of those through computational methods, I'm sure, that are described elsewhere and in the paper, you include both of those, and then you can have an understanding of what retrotransposon elements are being expressed in these two conditions.
So do they normally get thrown out because they're in such high numbers and we're really just looking for gene changes, or are there technical reasons? Or
my understanding is that some of it is technical reasons, in that at least in some cases, you can get sort of weird fusion products if the reverse transcriptase sort of starts reverse transcribing one of these products and then template switches, or sort of because of the repetitive nature of them, they're sort of technical reasons. And so I know of some other papers where people have done things like this and it's been a little weird. So one thing that I definitely, I certainly couldn't do, but I think that those computational things that Steph mentioned about the ways that you really carefully deal with these data are probably quite important. And if someone wants to do something similar, they should look into that, because you can't just randomly do it.
Right. So their comparisons that they highlight in figure 1 that they kick off the paper with are the splenic hematopoietic stem cells from pregnant mice compared with bone marrow HSCs from non-pregnant mice. Now, this was interesting to me because, it's going to help, I think, us talk about the remainder of the paper, because this particular comparison is made a lot. It's not that they found huge differences when they compared splenic HSCs in pregnant mice to splenic HSCs in non-pregnant mice. It really seems to be the biggest differences in gene transcription was splenic HSCs in pregnant mice to bone marrow HSCs in non-pregnant. And those are, you're dealing with now two different variables. And so you don't know if it's the unique nature of the HSCs from that organ or if it's the state of pregnancy. And I wanted to know what you guys thought about that, because they did do a lot of jumping in the comparisons, and depending on like which comparison made the biggest change is what was highlighted. For example, the changes that I thought were more, how do I want to say, you're only looking at one variable, non-pregnant versus pregnant spleen HSCs were in the supplement. So how did you all think about that? I mean, is that an appropriate comparison?
I think that, you're looking at a very different microenvironment.
Right.
You're trying to compare the two cells, and you're going to see a lot of differences, but whether those differences are meaningful and related to the question you're asking, I think is questionable.
Maybe that's the point, is that these splenic HSCs are just so unique, and we're going to dive into why during pregnancy. But it was a bit difficult to parse through these comparisons in RNA transcriptome analyses.
Did they have technical issues? Is that part of it?
So there's one assay that they discuss in a little bit called ATAC-seq. This is a way to look at chromatin access in the genome. And they mention in that paragraph that there were barely any HSCs in the spleens of non-pregnant mice to be able to do that assay. And so I thought, maybe it's a technical limitation.
Yeah, that's what I was wondering too.
They really don't bring that to you in the beginning. So
no, yeah. I remembered reading that, and I was looking for that when in discussing this figure, and I couldn't find it, but now you're right. They talked about it later. I don't know. I mean, I guess if you can get the samples, you compare what you can compare, but I don't know enough about HSCs and the, roles of the microenvironment of changing their function.
Right. So it could be technical. And I think what we can take away from overall from this first figure is that you see an increase in retrotransposons, including endogenous retroviruses and lines in the spleen HSCs from pregnant mice compared to the bone marrow HSCs from non-pregnant mice. So that's the biggest comparison. And that compares those differences were seen both at late and early stages of pregnancy. So
can you comment on, did you talk about this? What is considered a late and an early stage of pregnancy, and how that might be important related to this?
So day 14 was the first comparison. Mice give birth at 21 days. And so that would be considered, a week, middle, I think, of pregnancy. And then early for them was E7 to E9. So that would be after the males and females were paired, and then 7 to 9 days after that. So I think they could have done a late time point, but maybe it gets challenging when they get close to having their pups. You might lose some pregnancies. Maybe that was why they didn't do that. So that was interesting. And they talked about estradiol being a potential mechanism that would induce the HSC proliferation. And they did see that when they gave estradiol, they gave 2 micrograms per day for 6 days, and they saw a higher amount of HSC proliferation, and they saw a higher amount of erythroid precursor cells, which are the ones that develop into red blood cells. And so it's a potential way that's inducing this change. However, they didn't see the same number of retrotransposons that were upregulated compared to the pregnant mice. So it's not only estradiol that is likely progesterone. There's hormones and growth factor from the fetus that could be impacting. They also talk about other serial bleeding. They bled mice multiple times to try to kickstart their the production of more red blood cells, and they see similar increase in retrotransposons.
I was just looking at that exact section because I think that's the other piece that addresses the question that you asked a little bit, because they mentioned in the intro that serial bleeding, which you could imagine if you keep taking blood over and over again from a mouse, is also going to need to kickstart erythropoiesis, just like pregnancy. And so I feel like those serial bleed mice are their attempt to deal with that question that we were talking about, because these are non-pregnant mice.
Right.
They don't have the estrogen changes, and they're trying to see, do they get the same kind of changes? And there they do look at splenocytes and splenic red blood cells. But as you said before, this is all kind of supplemental data. Only a very small amount of it actually makes it into what you see in the paper. And so they mentioned that, oh yes, if we do this serial bleeding model, we get the same thing that we get during pregnancy. But I think I would have liked to see that a little more prominent to address those questions that you had mentioned.
Well, yeah. Do you think that maybe that a reviewer asked, is this just a pregnancy specific effect, or do you think there's something else? And then they did the serial bleed model, and then they incorporated that data in, because otherwise, why would you do the estradiol, right? Because if it's not dependent on that, because the serial bleeding is certainly not going to be dependent on that, then how are you getting at mechanism, right?
Yeah, I do think that they were trying to because when you look at figure 1, you're like, well, when you compare the same organ, HSCs, the differences are not as striking as when you switch organs. And so I think I agree. That could have been a way to prove that they see this in other ways that do not involve pregnancy. It's a great point. So they're setting up that when you have these type of stressors that require more blood, you have an increase in retrotransposons. And so they want to dive a little bit further into mechanism that I mentioned. They use this assay called ATAC-seq, which allows, it's a way to map the regions of open chromatin. And when chromatin is open, it indicates that DNA is accessible to transcriptional proteins. And while they saw little or no changes in chromatin accessibility in the bone marrow HSCs from pregnant compared to non-pregnant. Again, it was when you compared spleen HSCs from pregnant to bone marrow HSCs to non-pregnant. And those open areas of chromatin overlapped with retrotransposons. They say 28%, although that means there's 72% that overlap with other things. I didn't really know how to think when they say like 28%. I'm like, okay, great. Is that more? Or I wasn't sure how to conceptually put 28% into the paper, but basically when they do that in estradiol treated mice, they don't see the same level. They see similar findings of chromatin accessibility. So they're trying to prove this point that you have overlapping areas of accessibility where retrotransposon elements are. To build up this argument of what's going on. So the next thing they do, which is the more direct way to stop reverse transcription, and they give the animals antiretroviral treatment. These are called reverse transcriptase inhibitors. Tenofovir is one, and I'm sorry, the other one, pronunciating it, Emtricitabine. Who knows how to pronounce that? It's Emtricitabine. Emtricitabine. I think they use it also for other viruses as well. But both of these are used to inhibit HIV, particularly if you've heard of PrEP, which is pre-exposure prophylaxis. It is a pretty amazing drug that is able to keep the viral loads down because you're decreasing the way that those viruses replicate, which is the reverse transcriptase. And so if they are hypothesizing that it's the retro elements that are using reverse transcriptase to transcribe themselves and activate HSCs, if you treat mice, then that should reduce the effects they're seeing.
How much reverse transcriptase should we normally have? I thought it was 0.
It's pretty low.
I think it is low.
I didn't think we had that. Do we encode? Yeah. We don't encode a reverse transcriptase, do we?
We only encode it in these retrotransposons, although telomerase
yeah, right,
is similar, has some similarities.
Okay. So do these drugs act on telomerase?
Good question.
I don't know.
Emtricitabine, by the way, that's the pronunciation.
That's how you pronounce it.
Okay. Thank you.
Good question. Does it inhibit telomerase RT?
Because that would change how you interpret this, right?
Yeah.
Mm-hmm.
They're actively looking it up. But yeah, I was curious about these studies because, it's like, oh, okay, you take a non-retrovirally infected animal and then give them antiretroviral drugs, and now you see these things. And I'm like, well,
so the answer is yes.
It does.
It looks like it depends on the inhibitor.
Yeah, the NNRTIs particularly inhibit the telomerases.
Interesting.
Yeah, they could have specified that more clearly, right? Because that would have been just to tell your audience up front that these reduce the thing that we actually express.
In fact, the paper that I found looks like one of the specific ones they were looking at was tenofovir.
Interesting.
So that means you would age quicker if you're on antiretrovirals.
Which a lot of evidence in the HIV field is showing, because these individuals who had HIV and are now on an antiretroviral drug, they're thankfully aging. They're older, but they're more likely to get certain cancers. So it's not, they're not better off than people who are not on those drugs.
There's always a cost.
Right. So
that's why vaccines and preventing things are better.
Good point.
Yes. Although HIV vaccines, listen, we could take a vote of that virus is a beast.
Not great yet.
Another podcast for sure. So when they treated mice, to what we would expect if their hypothesis was correct is that when you treat non-pregnant mice, there were no effects on blood cell counts, bone marrow, spleen, hematopoiesis, HSC frequency, progenitor populations. But when you treat pregnant mice, you reduce the blood cell counts of red blood cells, but not white blood cells or platelets. You reduce these progenitors that can make megakaryocytes or erythrocytes, HSC frequency, division rates. And they did these cool reconstitution experiments where they were able to take HSCs from either pregnant or non-pregnant mice treated with RT inhibitor and put them in irradiated mice. And it was only in the pregnant mice, they actually don't even, the non-pregnant mice, there's no differences, that's in the supplemental, but the reverse transcriptase inhibitor reduces the ability of those donor cells from a non-irradiated mice to reconstitute themselves in an irradiated mouse. So
wait. When are they irradiating them? You're not irradiating pregnant mice, are they?
They're putting them in, they're taking them from the pregnant mice and putting them in non-pregnant irradiated mice.
Okay, got it.
Sorry.
I got confused by that for a minute.
Yeah, the donor cells
I've read
are from
this, but I didn't get that experiment
animals.
Clear in my head.
Yeah.
Got it.
So that's interesting. I like that experiment. I thought that was a solid piece of evidence that what their hypothesis could be true. And so what those experiments summarized, you can say that reverse transcription was necessary to increase HSC cell division, maintain and splenic, particularly splenic, not bone marrow, erythropoiesis, and maintain a normal blood cell count during pregnancy. So
the thought that just went through my head is, I wonder how long this effect lasts in the pregnant mice.
That's, I think, longevity experiments would be really interesting.
Yeah. So like is a mouse who has been pregnant and a mouse who has never been pregnant, will they be different in their hematopoietic and erythropoiesis processes?
It's interesting considering chromatin accessibility of these elements is dependent on things like methylation, which we know in other work, pregnancy does change methylation status. Whether those two things are associated, I don't know, but it'd be a good experiment, Brian. So the next thing they're setting up for us is, well, what is the mechanism? So now this is where we can now talk about C-gas and sting. C-gas is, I mentioned before, but shortened cyclic GMP-AMP synthase. It is activated by double-stranded DNA in the cytosol, which really isn't supposed to be there. And under normal conditions, the DNA is in the nucleus with mitochondria. And so the issue when retrotransposons, why they're able to activate it is through their life cycle, they are potentially bringing cDNA into the cytoplasm, whether that mechanisms of cellular stress or nuclear import or general genomic instability.
Yeah, I don't think they addressed it because there are other things that can activate the C-gas, including mitochondrial DNAs. So if you stress the cell and they become very metabolically active, for example, when they're proliferating to make new cells, they can then have some mitochondrial stress. Then that mitochondrial stress can release DNA, which can activate C-gas as well.
Yeah.
Which is a great point, because if we're going to critique this paper, the connection between retrotransposons activating C-gas and sting, because there are many things that can activate that pathway, and we'll talk about that, I think isn't hugely clear. There's one experiment they do, which I'll talk about, that links sting with reverse transcriptase activity. But that is a great point, Cindy.
Yeah, I mean, we're jumping ahead. Clear, they use C-gas knockouts. There's a role, but what exactly is the trigger is the question, I think. Yeah.
Right. But what's interesting, well, we'll talk about it. Because the C-gas knockouts, they actually never, they say that plays a role in pregnancy, but they never measure C-gas in the reverse transcriptase inhibitor treated mice.
Correct.
Only sting. So there's many things that activate sting. How do we know it's C-gas? I'm jumping ahead, but
I also will say everything you said, Steph, in terms of subcellular localization, is absolutely spot on.
Oh, I thought you said, I was going to say incorrect. I'm like,
no. Spot on. There is sort of a little bit of literature about C-gas and other DNA sensors acting on DNA that doesn't have proper chromatin structure, say that isn't bound on a histone. In the nucleus. And so again, whether this is in the cytoplasm, whether this is in the nucleus, just because the reverse transcribed DNA is not on is not properly chromatinized, is not something that they get at, but is also a level of pickiness of the field that is beyond where they are right here.
Interesting. Yeah. So this is why I love doing these podcasts, because you find the nuances in everyone's field and who's thinking about what. So, okay, so when C-gas binds to cytosolic DNA, it synthesizes the synthase and it activates sting. Sting triggers downstream signaling, a couple different proteins, and leads to production of type 1 interferons, pro-inflammatory cytokines. So they first looked at anti-phosphorylated sting using an antibody on HSCs that were sorted by flow cytometry from bone marrow and spleen of pregnant and non-pregnant mice. when sting or protein is phosphorylated, that is indicative that is activated. So it is a way to look at sting activation. These were interesting graphs. They were in the supplemental, not the main figure, but they show that they found higher levels of phosphorylated sting in the bone marrow and spleens of pregnant mice compared to non-pregnant mice. And when you treat the mice, the pregnant mice specifically, with reverse transcriptase inhibitors, you see reduced phosphorylated sting only in the spleen. So again, the spleen is unique. It's really suggesting this extramedullary erythropoiesis because the bone marrow, that was not true. Now, this is the last experiment, the last pieces of data we have on the animals treated with reverse transcriptase. What else would I have loved to see, considering we're talking about C-gas? Would have loved to see measurements of C-gas, interferons. The next section of the paper talks about C-gas and sting pathways and interferon production in pregnancy in general. It's assuming that the activation of that is coming from retrotransposons, but only this one experiment with the phosphorylated sting, do we link those two together? And I just would have loved to see more. Yep. Is that I don't know if
I would agree.
Yay or no?
I'm sitting here thinking about how to do that experiment. So yeah.
Yeah.
Yes. So really, was this one paper and it ended here and reviewers are like, we need to know mechanism? I mean, these could be really two papers because they're describing two different things, in my opinion, both equally interesting. Okay, but let's talk about what those other things are that they're presenting. So they go into fancy mice. We knock out sting. And I had to look this up because I didn't know what...
Golden ticket.
Golden ticket. I haven't worked with sting knockout mice, but when you look at it, it's sting and then it's GT #GT, and that's golden ticket. It knocks out TMEM 173, but why golden ticket?
I don't know. Bruce Beutler made this ENU mutagenesis colony of mice, and then each mouse strain after inbreeding had different defects, and they were really interested in all of the immune defect ones, but they had different things like missing eyes, curly tails, da, da, da.
Oh, wow.
They had lots of different mouse strains, but I don't know particularly why this one was called golden ticket, but they called it golden ticket. Maybe it was because it didn't respond to certain things and they were like, oh, that's, we won the ticket. I have no idea.
Oh yeah. Or somebody had just watched Charlie and the Chocolate Factory.
Maybe. Yeah.
So some pretty cool mice where you can knock out sting, and they found that sting mice had no effects. Sting deficiency in non-pregnant mice, no effects on all the things we've been talking about: blood cell counts, spleen cellularity, HSCs, progenitor cells, HSC division rates. But in pregnant mice, sting deficiency resulted in reduced red blood cell counts, not white blood cell counts or platelets, but red blood cells. Reductions in various progenitor cells, both myeloid and lymphoid and erythroid. And they did these reconstitution experiments like I described before. And both the bone marrow and the spleen cells from pregnant sting mice were reduced the reconstitution potential, I should say, were reduced in these sting deficient mice from both the bone marrow and the spleen. So this is interesting. We really were talking about a spleen specific effect. And now that you're knocking out sting broadly, now you're seeing this a problem in both bone marrow and spleen, which says this is probably just an interesting part of pregnant biology. How it's linked to the retrotransposons, that's a little bit less clear. So then they move on to understanding C-gas. And again, they didn't measure activation of C-gas in the reverse transcriptase inhibitor treated mice. But if you knock it out in mice, C-gas deficiency in non-pregnant mice had little to no effect. However, in pregnant mice, again, similar trend where you have reduced red blood cell counts, decreased frequencies of progenitors. And so it really suggests that C-gas activation and sting are important to increase HSC proliferation and red blood cell production in the spleen during pregnancy. They end up, as one would, if you're looking at the C-gas sting pathway, interferon production. Again, an assay, you take those reverse transcriptase inhibitor treated mice and you just look at levels of interferons, like they did here for their knockout mice, because that would tell you if it is due to the retrotransposons, you should see a decrease in interferons. When they looked at production of interferon alpha, they found not in the blood, but this will be relevant for the human studies I'm going to talk about later, so we'll remember this. Not in the blood did they see a difference in interferon alpha, but in bone marrow lysates and splenic lysates in pregnant mice compared to non-pregnant mice. And those differences disappeared when you got rid of sting or C-gas.
Yeah, I didn't know what to think about that, because normally when we think about interferon, it would be in the blood. So it's being made by cells in those compartments, but not secreted, or made at low levels, not enough to be able to reach a detectable level in the blood. It's unclear to me what that means.
I think it speaks to, there's oftentimes in the gut, we would find higher levels of cytokines and not in the blood. I think if you have more HSC, right. I don't know if they're claiming the HSCs are producing this.
Yeah, that was the other thing, right? If this is a sting dependent thing that's intrinsic to these retrotransposons being activated in the HSCs, they should be the ones making the interferon. That should be a doable experiment.
Yeah, totally. And that would make sense if the argument is that those HSCs are not in the blood, they're in the spleen and bone marrow.
Yeah. Which, by the way, couldn't they just go back to their original single cell data and see whether interferon and all of the stimulated genes were up in those cells?
Yes. Their RNA-seq data they go into next, they are now presenting us with all these different comparisons, sting deficiency in pregnant, non-pregnant. I think that's data from figure 1 that's being compared, reanalyzed in sting deficient mice.
Okay.
I think is the case, but that's a good point.
Yeah, but they just used whole interferon stimulated genes. They didn't look at interferon particularly. And I mean, they follow it up, right? Because they use interferon alpha receptor knockout. I'm jumping ahead a little bit, but it's just to show you that, it's dependent on this signaling feedback loop.
Agreed. They could have isolated HSCs from the spleens of pregnant, non-pregnant mice and put them in culture and see if they made interferon. Yeah. And so, again, because the comparisons were interesting in when we talked in the beginning, again, we're comparing two different things, spleen of pregnant mice, bone marrow of non-pregnant, but then as you get further into the paper, those restrictions get a little looser. Now in these experiments, we're talking about how sting deficiency affects both bone marrow and spleen. We're kind of decoupling what really they were trying to describe was the transposon-induced activation. So to Cindy's point, they also then look in IFNAR knockout mice, which they lack the receptor for interferon, and in non-pregnant mice, IFNAR deficiency did not affect all the things we talked about, blood cell counts, HSC frequencies, but in pregnant mice, IFNAR deficiency does lead to reduction of those things. So definitely that phenotype is true. This C-gas, sting, interferon pathway is required in pregnancy to induce HSCs, whether it's induced by retrotransposable elements, I think possibly. Yes, I especially I do like the experiments that they did earlier on where they transferred cells from pregnant mice, treated or not treated with RT inhibitor, and saw that impact. So they last but not least introduced some data in humans, which I can just say overall shows the diversity in humans and that humans don't follow the rules of mice, because there were so firstly, what they did is they did RNA-seq on enriched HSCs in pregnant women versus non-pregnant women, and looked at the lines and signs and saw that there were higher levels of these things in the HSCs from pregnant women than non-pregnant women. There was enhancement in interferon-regulated genes. they tried to look at retrotransposon transcription, but it was not uniform. Some women expressed them, some women didn't. And then when you went into pregnant women with HIV who were treated with RT inhibitors, you actually saw 2 of them had an increased expression of retrotransposon. Going back to what you all were saying, does that really reduce that enzyme in humans that's not the viral reverse transcriptase?
Yeah, I also kind of wondered in this one, they talk about their pregnant women, but they don't really talk a lot about, what stage of pregnancy these women were at. And you could certainly imagine that might influence this quite a bit. And they just, are like, hi, we had 11 pregnant women, and don't go into further data.
They did a little bit. I think it was week 11 to 35. So there was some. Was that just the HIV?
I think so, because then there were the last group of, I think I have to go back, 15 women where they compared.
I think this was just which were taking reverse transcriptase inhibitors. Yeah. So they all have to be the HIV.
Yeah, right. They would have to.
Yeah.
And they found that in those 15 individuals, so 15 who did not take RT inhibitors and 6 who did, they found that RT treated women experienced a decline in hemoglobin levels that led to anemia. So they then suggested that that's because retrotransposons are de-repressed during pregnancy in humans. So I think it's suggestive but inconclusive. It's hard with humans, low numbers.
Yeah.
So overall, I really enjoyed this paper. I think I got to learn a lot of new biology about pregnancy. I do think that retrotransposon inhibitor, RT inhibitor treatment did make a difference in their pregnant mice's ability to make red blood cells and activate HSCs. And separately, the C-gasting interferon pathway is important for HSCs in pregnancy. And future work should try to link those together. And what is the stimulus? They talked about estradiol, but it could be many things.
But it's interesting with the estradiol. We talked, a couple of episodes back about, people undergoing gender transforming therapy and taking, hormones. And I wonder how that's affecting, 1, their retrotransposons, and 2, their erythropoiesis.
Yeah. One thing that I remember from that paper, and sort of some subsequent looks at those data, is that paper really looked at testosterone and didn't look at estrogen. And if you remember when we talked to the author, he said, yeah, we actually did 2 cohorts and the data were so completely different.
Yep.
We couldn't put it into one paper.
Yeah.
And so part of me is still like, where's the estrogen paper? Where's the estrogen paper? and how much of this might show up there?
Yeah.
Yeah. And it would just be a matter of don't throw out those, retrotransposon elements and use fancy computational tools to make sure we're doing the right thing and then report back to us.
So I'm trying to figure out how. So CGAS, as you said, is a cytosolic double-stranded DNA sensor. So I'm trying to figure out how the retrotransposon DNA gets in the cytoplasm, okay? Because it's not supposed to be there. If you assume that the endogenous retrovirus is turned on, so you're going to make a messenger RNA, it's going to go out into the cytosol. It will be translated and make particles. I'm not going to do reverse transcription in the cytosol. That happens when the virus infects a new cell. So I don't know what's going on, right? They don't really talk about that. It's a virology question.
Yeah, that's why I mentioned there is sort of this idea of recognition of DNA that's not on a histone, potentially in the nucleus. And so there are some papers that talk a little bit about recognition in the nucleus based on the structure of the DNA. So that might be part of it. But the real question here is, why did the retrotransposon get turned on? It potentially is estradiol, or it's, in air quotes, some kind of stress. And how many of those types of stress also do things like lead to mitochondrial DNA release, which is known to induce mitochondrial DNA in the cytoplasm and cGAS. Obviously, that part doesn't tie into the reverse transcriptase inhibitor part of the paper at the beginning.
Right.
But mitochondrial DNA stress could be something that you could think about for the second half. And so what people can do is that sometimes people can actually fractionate the cell and get just cytoplasm and then do quantitative PCR to show that there is mitochondrial DNA in the cytoplasm. And so presumably you could take that same sample and then use primers to look for the retrotransposon DNA and see if it is also in the cytoplasm by those same methods. But I would be very interested here to see whether there was mitochondrial DNA in the cytoplasm in some of these stress conditions.
I was going to ask, how do you detect that? And the fractionation, it would be very nuanced centrifugation to get that out.
We're doing it here. So
nice.
It can be done. I mean, this happens in dengue virus infection, right? The mitochondrial broken, the DNA then signals.
Yeah, so. No, we have that. I've been doing that a bunch.
So that would be interesting, Brian. It's a full paper, but if they want to know what the signal is for cGAS STING pathway, that's one thing they could look at.
Yeah, absolutely.
Yeah, it just doesn't make sense that it's retroviral DNA.
Well, so isn't there a wrinkle to this, because there are a couple of papers that show STING can go to the nucleus, right?
Yeah, there are some papers that show STING and that show cGAS can both go to the nucleus.
Okay.
Yeah, so potentially detecting DNA in the nucleus.
Well, the nucleus is full of DNA then.
Right. Well, and that's where the structure becomes so important does the histone occlude recognition or is the DNA loose? And so I know there's a paper on some of this, I think from Steve Goff's lab, looking at histone carriage and how that blocks recognition.
Okay. Recognition by cGAS.
DNA sensors, yeah.
Okay. Thanks, Steph.
Yeah, sure.
I have one addition that I was able to look up while you guys were talking, and that is cool. Golden Ticket. So the AI overview of Google, so we'll take it with a grain of salt, says that it is from the Charlie and the Chocolate Factory, because when they selected these rare mutations, they represented a lucky find within the larger population of mutations that they found.
That's cool.
Yeah.
Guys have time for this one email?
Yes, of course. Let's do it.
All right, this is from Greg Kirchenbaum, who writes, I started listening to TWIV many years ago when I first learned about the podcast while attending ASV 2017 in Madison. However, I must admit I rarely miss an episode these days and listen with great interest, especially when the discussions pertain to SARS-CoV-2. Thank you to Dr. Griffin for the clinical updates as well. Today, after hearing you discuss this recent paper, along with your invitation for those with questions or comments to write, I decided today would be the day that I take the time to write and share my own comments.
Yay, good.
First, I want to offer my highest praise for Microbe TV and the great service you and your co-hosts, TWIV included, are providing through these podcasts. I, for one, really enjoyed the banter between co-hosts and the episode guests and the discussions of local weather conditions. I enjoy the discussion of data, whether only a brief snippet, Steph, your snippet was not so short, or a more detailed journal club-style figure-by-figure discussion. Second, I consider myself knowledgeable in the field of B-cell immunology, and specifically the literature demonstrating that circulating antibody titers, IgG in blood, wane over the course of months, either following SARS-CoV-2 infection or prime boost COVID-19 mRNA vaccination. To this end, I wanted to offer a small but important clarification on the terminology being used in this episode as it relates to B-cell memory. specifically a productive T-cell dependent B-cell response that leads to a fruitful germinal center reaction will yield both GC-derived memory B-cells, many of which are class-switched and possess somatic mutations in their IgH, IgL, and terminally differentiated antibody-secreting cells, plasmablast, plasma cells, so of which will take up long-term residence in the bone marrow or other suitable niches. As it relates to bone marrow resident plasma cells, the so-called LLPC compartment that was the topic of Nguyen et al. This is a paper we did recently. These cells represent wall one of adaptive humoral response, borrowing the terminology of Akaya et al., especially when one considers time points after the first few weeks when IgG produced by short-lived plasmablasts has declined, and we are considering the titer set point for a particular donor. Since IgG in blood has a relatively short half-life of 3 to 4 weeks, the maintenance of stable antibody titers in blood requires ongoing Ig production by antibody-secreting cells and continuous replenishment. The apparent waning of serum IgG titers in convalescent or vaccinated subjects has been well documented in the literature, is certainly in line with the data published by Nguyen et al. And co-authors in which spike-specific ASC are not entering the LLPC compartment based on their classification via surface CD19. However, as it relates to B-cell memory, there is also ample data, including data my group has reported, it provides a link, that confirms the generation of spike-specific memory B-cells at quite high frequencies in subjects, despite their low spike-specific IgG titers in blood. To my point, while the data presented by Nguyen and co-authors indicate spike-specific IgG positive antibody-secreting cells are not entering into the LLPC pool based on phenotype, it's important to note that natural infection or COVID-19 mRNA vaccination are both capable of eliciting robust memory B-cell responses. To echo Stephanie's point, circulating IgG titers and or the abundance of spike-specific IgG-positive antibody-secreting cells in the bone marrow does not provide any useful information on the level of neutralizing immunity conferred by IgA at the mucosal interface where SARS-CoV-2 virus infection first occurs. Moreover, if given the choice, assuming we are offered one, I would rather possess a clonally expanded memory B-cell compartment along with memory T-cells that are capable of re-engaging in an adaptive immune response and undergoing further affinity maturation via somatic mutation in a population of stable bone marrow resident LLPC that, while capable of secreting tremendous quantities of antibody for decades, are unable to refine their fine specificity and adapt to emerging variants, the ones that are responsible to reinfections. Said differently, while the apparent inability of spike-specific ASC elicited through mRNA vaccination to join the LLPC compartment is concerning, such cells only contribute to wall one of acquired humoral defense. In the ideal scenario, such Ig can and will prevent reinfection, but that is not our reality and a nearly unattainable bar to achieve for a COVID-19 vaccine or for seasonal influenza as well, or any vaccines, I would add. Instead, it's wall two memory B cells generated following a primary infection or vaccination that are most likely to impact the outcome of a future breakthrough. I know you don't favor this technology, infections, when pre-existing antibodies fail to provide sterilizing immunity. Returning to the paper of discussion, it remained to be demonstrated whether the bone marrow aspirates donors possess stable memory B-cell population. Maybe such work is already in the work and is certainly of personal interest. In closing, I really enjoy listening to the show. I look forward to potential future interactions with you and your co-hosts. I had the privilege of speaking with Cindy at AI24. I waved to Brianne as we passed each other on the escalator, and I'm still holding out hope that Stephanie will reinvite me to give a guest lab meeting at Case Western Reserve University. Yes, I called you out, Steph.
Yeah, I remember meeting Greg.
Oh, that was great. I'm really glad that he responded. Yeah, Greg is somebody with a very deep knowledge of apparent in this email. B-cell immunology, and in his mention of us, he has a really solid background in ferret B-cell immunology, which if you're studying influenza is really important. We don't have the reagents, but we're trying to get there. So
you should make some bespoke ones.
Yes.
Maybe some bespoke ones. Yes. But he brings up a great point. You know, what would we rather have? A pool of memory B and T cells that can adapt to different variants or strains, or long-lived plasma cells that are fixed in time to when they first saw their antigen. So I like that email.
I don't know though if that's actually what happens, right? Because, depending on when you're vaccinated, you have various outcomes with variants, right? So it's not clear to me that it happens at all.
You mean specifically those who are vaccinated only, or those who?
So he's saying we don't want this bone marrow fixed population of memory B cells that can only respond to whatever vaccination or infection induced them, right?
Yeah, I mean, he's saying that you don't want the plasma cells that theoretically are fixed, while the memory cells are not, because the memory cells can go back through a germinal sensory reaction, while the plasma cells have largely lost the B cell receptor and ability to be exogenously signaled, and so they're just going to keep pumping out the same old, and while the memory cells could change again.
And I think the paper we talked about, really you want both, and actually what immunity does is give you both, but the paper doesn't look at memory B cells, and so the the hang up on LLPCs, maybe it doesn't matter if they come back in their second paper and they show us, okay, memory B cells are there, it's fine. But Vincent, what you were saying is that the response to variants in individuals with memory B cell responses? I'm
just not clear how much refinement you get with the variant, right? Because there's this original priming effect, right?
Yeah, that's fair. And that's why, again, we talk about T cells.
Yeah, exactly. I mean, I do think that in a healthy person, they play a huge role, right?
Right.
Anyway, thank you for that, Greg.
So this is related to this waning antibody. Like everybody's making a big deal about waning antibody. We need to boost again. But that's going to be primarily produced through these long-lived plasma cells, right?
Yeah.
And it's okay if it goes down because, my understanding is we should still have memory B cells that can now get reinvigorated. And
after you get polio vaccine, your antibody titers wane, but they're still there very low because you have memory B cells. And then when you get challenged, they go up again, right?
With polio, though, it depends on whether you got the oral one, right? Because if you got the oral one, you would generate some IgA plasma cells that are going to park themselves right at the mucosal surface and be protective, right?
I'm not sure how protective they are. Think about this. You get the oral polio vaccine. Some people secrete virus for months. So if the antibody IgA were protective, it wouldn't secrete for months. It's kind of a fallacy, but with IPV, you have low antibody levels and they get boosted by a challenge.
Yeah.
That's cool. Immune 88. You can send questions and comments to immune@microbe.tv. If you like our work, please consider supporting us. Microbe.tv/contribute. Cindy Leifer is at Cornell University. She's also on Blue Sky and Threads.
Yep.
Thanks, Cindy.
Thank you.
Steph Langle is at Case Western Reserve University. Stephanie Longo, is that X still, Steph?
It is. I haven't ventured to the new platforms. Not that I'm against them. There's an activation energy to new social media platforms that I gotta go over.
Brianne Barker's at Drew University. She's also on Blue Sky. I don't have the right address here. Thanks, Brianne.
Thanks. It was great to be here. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Tatami. Thanks for listening to Immune, the Bostcast. That's infectious. We'll be back next month.